Hi guys, welcome to the last lesson of this block for physiology. It's on skeletal muscle. So before I get started, here's my office hours. You guys know I'm around on Sunday at this web address. It should be fairly easy to click and go. Of course, you can always meet with me by appointment. Don't study the old exam questions that I give you exclusively. Go through your notes, review your notes, but then after that, this is a good way to go through the material and actively study it and practice solving problems with it. Note that I have given you notes, and they are designed so that if you want to, you can just sit back and relax and watch this video and be fairly confident that everything you need to know is written down, and all you can do, you can just literally follow along figure by figure and just annotate here and there and highlight here and there if you want. Now, if you don't want to deal with the notes at all, that's fine too. You can watch the video and download the slides, which I have supplied for you, and simply take notes on the slides. That's fine. All the important information is in the video. You won't be missing anything if you don't ever look at the notes. On the other hand, those of you who like reading the notes and don't necessarily want to watch the videos, feel free to turn this off and delete it or whatever, and just read the notes, and that is perfectly fine as well. Everything's there, all right? And they will stand alone. The videos will stand alone. You can do this any way you want, all right? So, skeletal muscle. We're talking skeletal muscle. So, first of all, here's your muscle. Okay, and muscle makes up about half of your total body weight. Most of that is skeletal muscle. And it is made up of a bunch of cells, which are known as muscle fibers. We'll talk about that in a second, okay? Those bundles of those cells here, they have a connective tissue wrapped around them, and that connective tissue eventually sort of condenses down into tendons, which connect the muscle to the bone and provide the skeleton upon which the muscle hangs and acts. You guys know that from anatomy, if nothing else, I'm sure. So here is our muscle fiber here, okay? One single muscle cell is a muscle fiber, okay? And it is about 10 to 100 microns in diameter. That is different from cardiac muscle, which was about 10 to 20. So it's bigger, the skeletal muscle fibers run the entire length of the muscle, okay? So in length, it's the length of the muscle, tip to tip. So it is very, very long, and that is different from cardiac muscle. You'll recall that cardiac muscle is about 100 micron cells. They're connected on the edges by internal intercalated discs with gap junctions, and they act as a functional syncytium. That is not true here. This is one big muscle cell that runs the length of the fiber. Anytime that I mention differences from cardiac muscle, or you should take note of that. It is a traditional way to teach the material, and you will be asked about the differences between the two muscle types. It's just a good way to sort of focus on some of the more important aspects of how the functions of those two muscle types differ, because they are very similar in very, very many ways, as you will find out. The myofiber is made up of bundles of myofilaments, which are called myofibrils, okay? And those bundles are nothing more than sarcomeres in series. There's our sarcomere, they're in series here, lined up through the myofibril. Your sarcomeres are, of course, made up of thick filaments, which are made up of myosin, okay? And just like cardiac muscle, and thin filaments, which are made of actin, tropomyosin, and troponin, right here, okay? Thick filament, thin filament, all right? This is a quick histo review. I realize you've had it before. I'm not going to spend a lot of time on it, but I want to make sure we're all on the same page as I go through the lecture. The A band runs the length of the myosin. It is the heavy chain, okay? And it simply runs the length of the heavy chain. The I band is made up of actin. So actin goes from about right here all the way to the Z line. The portion of that, the light colored portion, is the I band from here to here on either side of the Z line. And this is actin which does not overlap with myosin. This is actin that overlaps with myosin. This is actin that does not on either side of the Z line. That is your I band. Your H band is part of the A band, okay, right here. And it is myosin, which is actin-free. So this is actin from here all the way to here. And then you've got some myosin in here, which is a little bit lighter colored, that doesn't have any overlap with actin. That is your H zone or your H band. And Z line runs here transversally through the myofibril. And it is proteins to which the actin anchors. So the actin goes here, it attaches to the Z line, and here it attaches to the Z line. And it anchors that actin and holds it in place. And your M line runs through the H zone, okay? And it is also a series of proteins that run transversally through the sarcomere and through the myofibril. And those proteins anchor the myosin and hold it in place, okay? So I had a slide that I showed in the action potential lectures, the synapse lecture, and I was going to show it here and I decided not to because this slide basically shows the same thing. That slide showed this nerve coming out of the spinal cord. You have an action potential which propagates down this nerve, which we reviewed. And it hits this N terminus here and initiates excitation in the neuromuscular junction, which is here, and we talked about that. So very quickly, the action potential opens N-type calcium channels, allows calcium to come in. Calcium allows the vesicles in this terminus, with the neurotransmitter, which is acetylcholine for the neuromuscular junction. It allows those vesicles to fuse to the nerve terminus, the presynaptic membrane, and exocytosis takes place. Acetylcholine is dumped into the junction. It diffuses across. It's a very small, narrow space. The top of these synaptic folds has acetylcholine receptors. The acetylcholine binds to those, and those receptors are also channels. And so the channels open, they allow sodium in and a little bit of potassium to come out. The net effect is depolarization of the membrane. You get an end plate potential. It hits threshold, and that opens sodium channels, and you fire an action potential which diffuses unidirectionally away from the synapse, okay? And don't forget that you have acetylcholinesterase at the bottom of these folds, and when this process is over and you want to terminate the excitation, the acetylcholinesterase destroys the acetylcholine, okay? So your action potential propagates away, and it propagates down into T-tubules. Again, very similar to cardiac muscle. And this is the T system here. The T-tubules, here's the cell membrane. The T-tubules are invaginations of cell membrane. And literally you have holes here, and this is all extracellular space in here. The T-tubule comes down and sort of wraps itself a little bit around the myofibril, which is here, okay? So here is, again, our transverse tubule, our T-tubule. In these two diagrams, it's pink in here. And what's inside this T-tubule is essentially extracellular space with calcium in it. And so what happens is your body has decided that in these big cells, like the skeletal muscle cells, it's much better to bring the calcium down to the myofilaments, because if you open channels in the cell membrane and they had to diffuse 10 to 100 microns into the middle of the cell where the myofilaments are, you're going to get a very slow contraction in the middle of that muscle fiber. And so by bringing the calcium down, or in this case, really bringing the voltage down in the form of the sarcolemmal membrane, you're bringing that voltage down into the middle of the fiber, and that's allowing SR calcium release to be initiated right on top of the myofibrils. So instead of, so the SR calcium is released like right there instead of being released sort of at the membrane and then having to diffuse in. Okay, so having said that, this is your sarcoplasmic reticulum, and it wraps around the myofilaments. There are a lot of sarcoplasmic reticulum calcium pumps here to take calcium up once contraction is done and you're starting relaxation. And though the sarcoplasmic reticulum has what is called lateral sacs, which come very close to the T-tubule, and where they become very close in apposition to the T-tubule, that's where the action takes place in terms of excitation, contraction, coupling. So one more thing to point out about this particular figure, the T-tubules actually run down and they hit this fiber and run along the border between the I band and the A band here. And you can actually see them in a microscope running in duplicate down the middle of this sarcomere, so that you've got calcium release on either end of this heavy myofilament, and that calcium is diffusing out from two spots right in the middle of the sarcomere where the action is. That's different from cardiac muscle. A skeletal muscle tends to be more specialized for contraction. There are little increases in efficiency here and there, and this is one of those. Cardiac muscle, the T-tubule comes down along the Z line and hits the fibers at the Z line. Here you've got two that actually, you've got them right where the action is, running right down in between the two Z lines instead. So a little bit of a change, another little difference between cardiac muscle and skeletal muscle. So here is your lateral cisterna, your lateral sacs of SR. Here is your T-tubule. Your action potential is coming down the T-tubule, okay? And it is exciting the membrane here, and see where the lateral sac comes right close to the T-tubule. This is the T-SR junction, and this is where all the action takes place for excitation, contraction, coupling. So in the T-tubule membrane, the sarcolemmal membrane, the cell membrane, we have these voltage sensors or depolarization sensors, also called DHPR or DHP receptors, because they bind to hydropyridines, which, as you guys know, are calcium channel blockers. And these are, in fact, calcium channels, but you can ignore that, because the calcium influx when these open up is very low, very small, and very slow, and does not contribute significantly to contraction. So you can basically forget about the fact that they're actually calcium channels. Their job is to change conformation when the voltage comes down, and I'll get that in a second. Here we have ryanodine receptors and the sarcoplasmic reticulum membrane. Those, as you'll recall, are SR calcium release channels, and your calcium release is going to obviously take place through those channels. So your action potential comes down your T-tubule membrane. The voltage hits the DHPR, or the voltage sensor, or the depolarization sensor, and this sensor changes conformation in response to the voltage. And this voltage sensor is directly hooked up, at least that's the current predominant theory, and it's very, very well accepted in most scientific circles. It's directly hooked up to the SR calcium release channel. So when this changes conformation, because these two are basically hooked together, this also changes conformation, and you get calcium release, because that change in conformation opens up the calcium channel. So the voltage here, sensed by the voltage sensor, causes a conformational change in the ryanodine receptor, which opens up the channel, and calcium comes out, and it's this calcium that is released and causes contraction. And that's known as a mechanical mechanism. It's the mechanical mechanism for excitation, contraction, coupling. Mechanical because these two things are hooked up, and this mechanically opens this receptor to allow calcium to come out, okay? Note that that release mechanism is not the same as calcium-induced calcium release, which is the process by which calcium release is initiated in cardiac muscle. In skeletal muscle, calcium release is initiated by voltage through the mechanical mechanism. In cardiac muscle, it is initiated through calcium-induced calcium release, where calcium, significant calcium, actually comes in and accumulates in this junctional space, and the calcium binds this ryanodine receptor, and it's the calcium binding that opens this receptor. That is not happening here. That's cardiac muscle. Here, this is a direct connection, and this directly opens this. Calcium influx is not necessary and plays no role in the process of initiating this release, okay? So in summary, we have an end-plate potential generated at the neuromuscular junction by acetylcholine release from the presynaptic membrane. We have that triggering an action potential because this is designed to always hit threshold with much space to spare. And an action potential is fired. It propagates into the T system where the DHPR changes conformation and directly opens the ryanodine receptor, and calcium is released from the SR. All right, so that's our first video for this lesson. And in the next 2 lessons, we'll go over how contraction takes place. Again, you're going to find that to be very similar to cardiac muscle. And then we'll go over how force of contraction is altered in the third and last video of this lesson. All right, thank you for your attention.